After understanding about the basics of biotechnology, that is what does biotechnology mean, let us understand the principle that is involved behind all such biotechnological processes. In order to understand the principles of biotechnology, mainly two techniques occur which have given birth to such modern biotechnology. First, genetic engineering, and second, maintenance of sterile condition. So let us begin with the first, that is genetic engineering. Genetic engineering cannot be considered as a constructed part because it is a wide part of biotechnology. That is, it is the major part that constitutes the biotechnology. Genetic engineering can be defined as a term that refers to the manipulation of nucleic acids like DNA or RNA of an organism. Thus, by a simple or complicated cross combination of such DNA and RNA of different organisms, either of the same species or different species, in different manner can give rise to the genetic engineering, with the help of which various products for the welfare of human beings can be obtained. Now, the organisms which take part in such biotechnological processes in order to perform the genetic engineering processes are referred to as genetically modified organisms, and therefore organisms whose genes have been artificially treated or artificially altered for a desired effect can be defined as genetically modified organisms. Which is also named as GMO. In organisms that exhibit asexual reproduction, preserves their genetic information. As we all know that the reproduction process in such asexual organisms is comparatively easy in order to understand the reproduction method because the genetic information within them is preserved and comparatively easy to understand than the sexually reproducing organism. And therefore, in sexual reproduction, provides, that is those organism provides opportunity for various type of variations and formation of new combination of genetic aspects. That is new type of genetic combination, which may somehow be beneficial or helpful to other organisms. Thus, this whole study involves genetic engineering and its applications. In this technique of genetic engineering, the transfer of nucleic acid molecule, that is either DNA or RNA, occurs from one cell to the other. Now these cells can be either of the same organism or different organism. The cell here where the transfer occurs, that is from where the DNA is extracted, that cell is called donor cell. That is the cell from where the DNA or RNA is extracted. And the cell within which this extracted DNA is inserted is known as recipient cell. Thus, recipient cell can be defined as the cell into which the extracted fragment, that is the fragment can either be of DNA or RNA, from donor cell is inserted. Thus, in this technique of genetic engineering, the desired DNA fragment is isolated from a suitable donor cell and is transferred to a recipient cell. This inserted DNA fragment cannot multiply, that is, will not be able to execute its action unless it gets integrated into the genome of the recipient. Therefore, the recipient DNA cell that possesses a specific sequence initiates the DNA replication. Such specific sequences of cell which possess, that is, which is possessed by recipient DNA is known as origin of replication. Thus, from this origin, the DNA replication begins. As a result of which, the integrated DNA fragment replicates and multiplies itself in the recipient cell. Such a method of genetic engineering in which the replication of DNA occurs within the recipient cell is known as cloning. Thus, cloning can be defined as a technique of genetic engineering in which the DNA fragment is transferred from a donor to recipient cell of suitable size starts replicating or initiates DNA replication within the recipient cell. As said earlier, in order for the initiation of this DNA replication, the sequence, that is origin of replication, is very essential in order to carry out the replication process. The second most important aspect of genetic engineering that comes after cloning is recombinant DNA technology. Recombinant DNA technology is also a type of genetic engineering technique like cloning. The basic principles of such recombinant DNA biotechnology includes generation of DNA fragments, and from it, the selection of desired piece of DNA. The second point that comes after it is insertion of this selected DNA into a cloning vector which is generally in the form of plasmid, which helps in the creation of recombinant DNA. The third point that comes after it is the introduction of recombinant DNA DNA is generally in the form of vector, so recombinant vector into the host cell. Following which the multiplication and selection of clones occur. that contains the recombinant molecule. Following with the multiplication and selection, finally the expression of the clone appears. That is, the expression of the gene occurs, which results into the formation of finally desired product. That is, this expression forms the desired product. Thus, this is all about the basics of genetic engineering, which involves techniques like cloning and recombinant DNA technology. Now, after understanding about the process involved in genetic engineering, let us understand about the advantage of such a technique. That is very obvious that if such processes are involved within it, there must be various advantages of it. In this technique, during the isolation of desirable genes from the target organism is done, at this time no undesirable traits or undesirable genes gets mixed with it. And therefore a pure and proper product along with desirable characteristics can be obtained easily with the help of this technique, which is unlike traditional hybridization method where along with desirable genes, sometimes undesirable genes also gets mixed up, which results into the formation of impure product and thereby incomplete process. This is all about genetic engineering. Now let us understand about the second principle, that is maintenance of sterile condition. Now it is very essential that if such genetic engineering techniques are performed, then the conditions under which these techniques are performed should be sterile and in proper manner. That is, depending on the process and the organisms that are involved within it, constitutes the sterile condition accordingly. And so, the conditions in chemical engineering processes in order to enable the growth of only desired organisms that are obtained through the technique of genetic engineering to obtain large quantities of various substances like antibiotics, enzymes, hormones, and many others. Can be done with the help of genetic engineering technique and other biotechnological processes, but only under specific and sterile conditions. With these all mainly includes the principles of biotechnology.